morning. I'm Amber Salzman, also on the ALD Connect board, and I'm tasked with trying to keep us on time, and then they really challenged me by starting late. So, with that, I'll introduce people and try to manage the questions and such. But again, we have breakout rooms. This is an opportunity to catch people in between and such. We want to make sure that people's questions are addressed. So with that, let's kick off the meeting with Dr. Florian Eichler, who's going to talk about ALD in the era of precision medicine, the opportunities and challenges. For those of, everyone knows him, but I will introduce him anyway. He is the co-founder and president of ALD Connect, and he's a professor of neurology at MGH and Harvard Medical School.
Wonderful to be here, and thanks, Amber, and a big shout out to Kathleen and Kelly. And I think before I forget it, we should have a round of applause just for everything Kathleen and Kelly have done here. So what I tried to do today is give here an overview of where I think we are with precision medicine and gene therapy. Recognizing that this is evolving and in flux, and there are many exciting aspects to this, but there are also significant challenges. So here, briefly, my disclosures. Now, I'm also happy to see many people who are new to this community and new faces, so I don't want to start by just assuming everybody knows what ALD is and what adrenoleukodystrophy stands for. So just a slide or two on the relationship between the single gene and the different manifestations. So X-linked adrenoleukodystrophy is caused by variants in ABCD1. This is a gene that encodes HAF transporter in the peroxisome and needs to either couple with itself or another HAF transporter to import very long chain fatty acids into the peroxisome. So all my work has been in the past 10, 15 years really trying to figure out ways to correct this gene and address the problem, so to speak, at its root. But recognizing well that even knowing of these variants, we cannot predict what kind of manifestation you will experience over time. And this ranges from adrenal insufficiency to cerebral ALD affecting the brain, and axonal degeneration of the spinal cord, also known as AMN. So just as a brief overview, you're born with a gene defect, and as you have this gene defect, you also have elevations in very long chain fatty acids that puts you at a higher risk during early childhood between the ages of 4 and 10 to develop cerebral ALD. If that doesn't happen, then going into adulthood, you are at highest risk neurologically for the spinal cord form of the disease. Very important to note that you still remain at risk for brain disease, and as an adult it's important to continue to get yearly brain MRIs to monitor for that. You note here that we've also recognized over the past 10 years that women are not just mere carriers, but also develop symptoms over time. And these are similar to the symptoms in men with AMN, with the difference that women do not develop cerebral ALD and do not develop adrenal insufficiency, but they can develop the spinal cord disease similar to men. One thing I'm pointing out here is that the different manifestations in brain as well as in spinal cord also have different forms of pathology, one leading to inflammation in the white matter of the brain, and the other having this long tract spinal cord degeneration. And that's going to be important as I try to point out over time how to use different forms of gene therapy to address these different manifestations. So when we talk about precision medicine, what do we actually mean? Precision medicine is a form of targeting genes, and that can range from adding genes, silencing genes, and now in this day and age, editing genes precisely. But it also implies delivering that cargo of new genes or the gene editing machinery in different ways. And here you see a little overview of the delivery methods. And I'm excited to say that several of these are already in human in ALD. So you have on the one hand AAV-based in vivo gene therapy, which is delivering directly to target organ, in case of ALD directly to the spinal cord in adults. That is now a trial underway. And then ex vivo gene therapy is now become an FDA-approved drug that is a modality where you have to harvest cells from the bone marrow, transfect them outside of the body, and then deliver those cells back. I'll speak briefly about that. And you'll hear later on a little bit more about new modality called antisense oligonucleotides, which is actually not directed at DNA, but at RNA. So if you think about your genetic code, your blueprint for your cells, the DNA encodes RNA and then protein. So you have different ways of intervening, and I'm just going to start off by highlighting really work over 10, 20 years that occurred with Christy Duncan, David Williams, Patricia Mussolino, many others here in this room, who helped build on the experience of bone marrow transplantation and recognized that while bone marrow transplantation was effective, it had downsides with engraftment problems, graft versus host disease, and we could improve upon that by taking cells from the boys themselves who had inflammation in the brain, harvest those cells, transfect them with a lentiviral vector, carrying a healthy copy of the gene, and then giving those cells back after a brief amount of myeloablation, chemotherapy using either cyclophosphamide and busulfan or fludarabine and busulfan. And then we watched these boys over a 2-year period in this trial, and the cells move from bone marrow to blood and from blood to brain. This is very similar to a normal bone marrow transplantation, with the difference that these cells are coming from the boys themselves. So in September of 2022, that became the first FDA-approved gene therapy for leukodystrophy. And I think we were all very excited about the progress. What we have recognized is, while there has been amazing outcome in terms of neurological stability, there's also been setbacks with the occurrence of myelodysplastic syndrome, a form of cancer of the bone marrow. And this occurs in about 10% of the boys who were treated. And here papers that Christy Duncan and I and others here published last month to highlight both the amazing progress we've made, but also the setbacks in the field. And I think this is part of the message today, is that as we are making progress in precision medicine, is sometimes 2 steps forwards and 2 steps and 1 step back. If you see ex vivo lentiviral gene therapy for childhood cerebral ALD as an opportunity to build on bone marrow transplantation, recognizing that there are certain cells in the brain, the microglia, that need to be corrected, that the blood-brain barrier is open, that you can, by using autologous cells, improve upon conventional bone marrow transplantation. That has borne out, and that has shown to be effective, with 94% of the more than 70 patients to date achieving stable neurologic function. But with the 10% of the children having myelodysplastic syndrome, we still have a lot of work ahead and need to make progress to understand how and why the insertions of the lentiviruses are causing this manifestation. You will hear more from Dr. Prasad this afternoon about this, but just to summarize here briefly the occurrence in the 7 boys, and to highlight that 6 of these 7 who either had myelodysplastic syndrome or another form of cancer called AML were treated with one form of chemotherapy called fludarabine, which is not as strong as the cyclophosphamide used in the first trial. And it might be that weaker form of chemotherapy is contributing to the myelodysplastic syndrome emerging. But I want to highlight we do not understand all factors contributing, and there are many efforts ongoing to improve this. What are those things that we want to improve upon? I think now I want to switch a little bit into future mode and speak a little bit to what I think is going to happen in the coming years. Here's some work highlighted by David Williams at Boston Children's, who has been instrumental in designing this trial for ex vivo lentiviral gene therapy, and is now looking at new vector constructs with new promoters, recognizing that we used a very strong promoter initially in our trial, an MND promoter. He is now looking at constructs using PGK, HLA. Suffice it to say that this is the cargo gene we're delivering, and there are various different elements that go into vector constructs, and the promoter itself is what drives overexpression of the gene in many cases. These are weaker promoters compared to the ones we use, but this one, for instance, the PGK, was used in metachromatic leukodystrophy gene therapy trial. I lost my only screen. Okay, I will try to be as explicit as possible, and no. The future is uncertain. So let me try to craft more of a narrative as I try to make this clear, and Dean, I'm sure, is trying his best to get the screens back up and running. So, could overexpression have contributed to more insertion of the lentivirus into the genome, inserting into certain proto-oncogenes that have given rise to myelodysplastic syndrome? It is not completely clear, as we see insertions across the entire genome, and many of the kids have remained completely stable even with those insertions. But it is safe to say that if we had a weaker promoter, it's possible that it would lead to less insertions and therefore be safer. What we need to balance this out with is making sure that we still achieve efficacy, and there's reduction in very long chain fatty acids. The other pieces you're not seeing here is efforts around gene editing that Ben Kleinstiever is undertaking. So beyond thinking about an exogenous promoter, you can think about preserving the promoter that your cells already have. We call that the endogenous promoter, and simply performing editing to precisely achieve a nucleotide switch of that specific variant you have. And often that with CRISPR-Cas9, many of you might have heard of this, you have opportunities to do this, but this often leads to double-strand breaks in DNA that could bring added burden of insertions or deletions. So Ben Kleinstiever, working with us, has been really pioneering some new technology around base editing, and which allows for a switch of the nucleotides without double-strand breaks. And we recognize that switching out for every individual mutation, knowing that there are more than 1,000 different mutations across the 10 exons in ABCD1 is very cumbersome and would require us to have a guide RNA for each single of these. So what we're now thinking of and planning is a broader approach that allows us to do what we call template jumping, which allows us to insert a much larger sequence from exon 2 to exon 10 into the cDNA of patients. And we do this in a two-step process. The first step is using something called click editing to go into a non-coding part of the DNA in intron 1. We put in something called a landing pad, and that allows us then to insert on top of this, the exons 2 to 10. Just to highlight that we've now been able to achieve this insertion of the landing pad, and here in kidney cells, HEC293 cells, you can see 40% efficiency in editing. We still are achieving about 5% in fibroblasts, so this is rapidly improving. At the same time, we also have to make sure we're not getting other edits, bystander edits, or off-target edits. So I just want to highlight that beyond vector improvements and working on the promoter, there are also opportunities to exploit gene editing to become even more precise. But the next question is, how do you target the right region in order to perform precise editing? And there are differences here between ex vivo and in vivo gene therapy. And I'm going to make an argument that you need in vivo gene therapy in order to get to the spinal cord for AMN. And this is just briefly highlighted by work pioneered by Yi Gong, Casey McGuire, my lab, and then that was then carried forward with Swan Bio and now with Spur Therapeutics. But here the early proof of concept, you can add genes by using an AAV9. You can lower very long chain fatty acids in spinal cord of the mouse model of ALD. And then this was then taken further in a double knockout, ABCD1, ABCD2 double knockout, that showed that you also here can add ABCD1, lower very long chain fatty acids, and also get correction in grip strength and hind limb clasping in these mice. What I wanted to also show is some work here in non-human primates. Before you go into humans, FDA requires that you test this in a larger species, and here we were able to show that comparing slow, a brief bolus infusion into the spinal fluid compared to a slow infusion, you got much better delivery to different parts of the spinal cord with a slow infusion over 24 hours. This was then further optimized so that now this is in human trial with slow infusions into the spinal fluid over 4 to 6 hours, and is currently enrolling at UMass Worcester in Massachusetts, and as well as in Amsterdam. And I think Mark Engelen is here and will be able to also talk about this later. So what can we do for women? What are the strategies to restore ABCD1 function in women? And I think it's an exciting topic to think about because really keep in mind that as opposed to men who only have one X that carries the defective ABCD1 gene, women have two X's, and during early development, one of the X's usually gets inactivated in order to balance out dosage of genes, and one can exploit that mechanism to reactivate the healthy X in the case of women with XALD. Here's Jenny Lee, an world expert on X inactivation at MGH, shown here, who's collaborating with us to achieve this partial reactivation of the inactivated X chromosome. And we do this by targeting a certain sequence called exist with compounds that disrupt RNA structure and exon activation. As proof of concept that this is important, I just want to highlight a few cases in our clinic of women who had 100% skewing either towards the healthy X or towards the unhealthy X. And as you can see, it says 100% skewing, and in one case you have a lot of ABCD1 protein, in the other case you have no ABCD1 protein, showing that really that skewing can go either to the healthy X or the unhealthy X. Here in the middle you have a balanced case of 54 to 46% skewing. And so in fibroblasts of these women, we showed that you really had complete suppression of ABCD1 in one case versus really normal levels of skewing that's the healthy X. And this results in high very long chain fatty acids of ABCD1 of very long chain fatty acids in the patient who was skewed in the right direction. So we've gone on to now create a mouse model that allows us to model this. So looking at T6 knockdown induces skewed exon activation in female ALD mice. This T6 is what controls the activation process of the X chromosome. So anytime when a woman is born, the early development of that, the cells have already gone through a process of inactivating one of the X's, and we can in a model cross ABCD1 with the T6 knockout, and you can see complete suppression of ABCD1 in these mice. And these mice have levels now of very long chain fatty acids that are as high as the male mice. And so really this model, for all intents and purposes, has created a severe phenotype similar to men, and allows us now to investigate reactivation of the healthy X. So more to come. I just want to highlight that beyond ABCD1, there are other genes that also influence adrenoleukodystrophy and can influence how ALD manifests. Remember, ABCD1 sits here on the peroxisomal membrane, imports very long chain fatty acids into the cell. It can also be compensated for by other genes such as ABCD2. We know that the very long chain fatty acids themselves are elongated by ELOVL1, so targeting that a gene would also be very exciting. Looking at SED1 that contributes to saturation of these very long chain fatty acids is another possibility. So ABCD1 expression, however, is very cell type specific, and we have to think about that and recognize that in some cells there's much higher ABCD1 than in other cells, and in disease states this may change, whether you have cerebral ALD or you have spinal cord disease, and in what cells ABCD1 is expressed seems critical to me for precise editing and precise targeting. So we find that actually there are regional differences in neuronal ABCD1 expression. If you look at brain, this is a normal brain, and you see high ABCD1 in the basal ganglia, hypothalamus, and deep gray nuclei, but not actually in motor cortex, which is a little surprising given that you see such degeneration of the motor tracts in AMN. So I'm always, since seeing this, been wondering whether this influences neurotransmitters in this brain disease, and I'm particularly interested in this as women with ALD not only have displayed movement disorders I've not seen in men, but I've also found them to have more prominent restless leg syndrome. And I think it's important to understand that leg discomfort in women it can arise from a variety of different things, from spasticity, neuropathy, vascular insufficiency to restless leg syndrome. And we have a first trial here that's looking at leg discomfort in women with ALD. And there are two phases to this. The first phase, we are really, we've enrolled, I think, close to 70 patients now who have been surveyed and screened as to the nature of their leg discomfort. And then if they screen positive for restless leg syndrome, they go to a phase 2 where they have a crossover study looking at a drug that influences dopamine. And I think we've seen some amazing successes with some women really pointing to the fact that neurotransmitter pathways could play a role here in how leg discomfort, gait activation, and balance play a role. Okay, I'm at the end, and this is really a first FDA-approved therapy for ALD. In ex vivo gene therapy, we need to balance the emerging risks against efficacy. We have to improve vector design and gene editing that is currently underway. In vivo gene therapy has led to a first trial in AMN. Women are still lacking trials in precision medicine, but skewed exon activation can modulate disease. There are many more genes in the lipid pathway that can be targets of therapy, and it's a very exciting time. And I think we have a lot to look forward to. But precision lies not only in the genes, it also lies in the symptom etiology and defining symptoms carefully that can lead to new trials. So sorry about the technical difficulties, but I hope my message still came across. Okay, thank you.